the title of it, Life, A History of Mergers. So I'm going to set up what I think the issue is and how we're going to talk about it. There are Hollywood movies called The Tree of Life. There's the Tree of Life. I think there's a beauty products called The Tree of Life. The Tree of Life is everywhere. I'm going to say that there's a little bit of a problem with The Tree of Life. I'm not the first person to say that, but hopefully today I can outline what the issues are and so on. It's not a minor tweak, it's a major thing. The Tree of Life, I'll take us through it a little bit, is an hypothesis. And any good hypothesis should have a variety of things associated with it. It should be able to explain the thing that it says it's able to explain. So that's one. It should be robust. It should be generalizable, and so on. We should be able to take a particular hypothesis, and if we think it's correct, its predictions should be obvious to us. And what I think I'm going to say is that that the Tree of Life hypothesis doesn't quite do a good enough job, and hopefully at the end of this hour you'd agree with me. But if you don't, there's wine afterwards and we'll all be fine, and nothing too bad's going to happen here, probably. And if you know what this is, it's one of the depictions of the Scala Naturae, the great ladder of life. And you can see at the very top of the Scala Naturae was God. And at the very, as you come down, there's angels and saints and so on. Archangels are above angels, all the way down to the earth is at the bottom, water is above that, and so on. This was a very, very serious idea for about 200 years, that in reality, the world conformed to a great ladder of life. Maybe if I talk a little bit about philosophy, there's two things that you could think about quite often. What do we study? Well, we only study things that we think exist. So in philosophy, people talk about ontologies, and then they talk about epistemology. Epistemology is the way in which you derive some knowledge about that thing that exists. We only study things that we think exist. So for a long, long time, people thought that the Scala Naturae existed. It was a natural thing, that there was a ladder of life, and the object of study should be to try to figure out what went above what and what went below. And of course, lions were decided to be the king of the jungle, and so they're at the top, and so on. It got into all aspects of life, because this was considered to be the natural order of things. It justified monarchies, it justified racism, it justified sexism, it justified all kinds of organizations of society. In the world that we work in biology and so on, it sort of encountered a few problems, as you might imagine. I mean, there were some organisms that, they arbitrarily, I think, from today, but putting the lions above tigers and so on was considered to be, correct at the time. But some organisms, there were problems with it. Hydra. What was a hydra? Was that a plant? Was that an animal? At the time, people didn't have very, very good ways of trying to figure these things out. You had lots of arguments about this. And this went on for about 200 years. And slowly but surely, people figured out that you couldn't really organize all of life according to this Scala Naturae. And it sort of began to fall out of fashion. And even though Linnaeus, who's got a little statue up on the turn of the stairs, Linnaeus sort of produced a 3-volume treatise on the ladder of life at one stage, but it was at a time when the ladder idea was in decline. It wasn't a great idea. The reason I mention it is because ideas sometimes they don't last. Big ideas don't last. They're quinarians. Have you heard of the quinarians? They believed in the number 5, and that the world was organized according to the number 5. That lasted a while, and it sort of died out. But the Scala Naturae, we don't hear much of it now. Although I will say that when I was at the Natural History Museum, I was in the division of lower vertebrates, which is it's actually lower vertebrates in mathematics, would you believe? But anyway, but there was still sort of a little bit left over. I'm not sure what this was ever called, the great asparagus bunch of life, maybe. I'm not quite sure. But this is the idea that different lineages sort of existed beside each other in some sort of way, and that was what life looked like. And then eventually people got to these sorts of ideas here. These are varieties of hyacinths, and these are nested groups within groups within groups, something that's very tree-like that we see today. And the tree-like idea really did supplant the ladder idea, and for very, very good reasons, because you didn't have to put things above and below each other that could be beside each other, and that sort of worked very, very well. It also had a lot of supporters. This is Darwin. It's a very famous, I think, drawing that looks a little bit like a fucus vesiculosus. And he said, I think, and he made this drawing, and lots of people latched onto this and thought this is a great idea. He wasn't the person that came up with it. Trees had existed a long time before Darwin, but he sort of said that he liked the idea of trees. He also liked the idea of the coral of life, with the living systems growing over and on top of dead systems. But quite often you see quotes like this from Darwin. It's a really good quote. He said, the affinities of all the beings of the same class have sometimes been represented by a great tree. I believe this simile largely speaks the truth. The green and budding twigs may represent existing species, and those produced during former years may represent the long succession of extinct species. At each period of growth, all the growing twigs have tried to branch out on all sides and to overtop and kill the surrounding twigs and branches in the same manner as species and groups of species have at all times overmastered other species in the great battle for life. What actually happened around that time was the establishment of the tree-likeness of life. And the tree is a great metaphor. And it's very interesting because Darwin was really, really, really great at using metaphors of all kinds. He's pigeon fancier and all of this. And he used metaphors continuously. And the idea of a twig of a tree being metaphorically like the species of all living things, that was very, very good. It's very, very persuasive argument. And not just that, but it sort of worked for the zoologists and the botanists in particular. And the idea from then on was, how do we get data? How do we make trees? Methods, data, and on and on about. The problem started when the microbiologists got involved. And this is C.B. Van Niel. I'll read this in a second. Sorry, it's the last of my reading. But C.B. Van Niel, very, very prominent Dutch-American microbiologist, very, very interested in the classification of bacteria. And that was really, really important to try and classify bacteria because we wanted to know what they were, the same as the zoologists and the botanists. But try as you might, you couldn't do it so easily. So if you had some bacteria that were motile or they were gliding, and you tried to put them into a group and you looked at some other aspect of physiology or biochemistry, like cell walls and so on, they didn't fit together. Like if you think about sort of, mammals, if you think about those, they have a lot of similarities, lactation, single bone in the lower jaw, hair, then there's a group within the vertebrates. You start including birds and fish and so on. And then the vertebrates will be within the animals and so on. So groups within groups within groups, that kind of natural order. Very, very easy to see from animals because they had such a diversity of characteristics. And there was a certain amount of congruence across these characteristics. Bacteria, not so much. C.B. Van Niel was initially very, very positive about this. And up to this paper in 1946, prior to that, he'd written lots of papers about, we can classify bacteria this way or that way using this characteristic or that. 1946, he'd sort of given up. 5 years before, he wrote a very optimistic paper in 1946. He wrote things like this. So far, the accomplishments in this direction, trying to come up with a system of classifying and explaining the evolution of bacteria. The accomplishments in this direction are meager, and that much of what has been done has gone under the name of taxonomy and has pretended to be a contribution to our understanding of natural relationships of bacteria is in reality no more than a simple device useful for the construction of a system of classification which permits the ready recognition and identification of a bacterial culture. Animals and plants could go on the tree of life. They were welcome there. They fitted. Bacteria, not so much. They were sort of put into a filing cabinet and they were taken out whenever you got sick and we figured out what was wrong with you. Because, I mean, let's face it, if you had a chest infection, you wanted to know if that was tuberculosis or influenza. You didn't really care about its evolutionary history. You just wanted to know what it was. So bacterial classification and evolution went through a period for about 50 years where it was completely in the doldrums. Nobody could figure out a way of coming up with a decent classification system. There was a book called Burgess' Manual, and it really wasn't trying to explain the evolutionary history of bacteria. It was just a key to try and describe them and try to identify bacteria. Van Niel went on, he said, it's not hereby implied that such systems are inferior to the natural systems of the botanist and zoologist, but merely that they serve a different purpose and are arbitrary. If they could be more generally recognized as such, instead of being regarded as inviolable expressions of natural laws, it would be easier for specialists with divergent interests to reach a mutual understanding. He said, look, all we can do with these is just describe them, and that's enough, and that's it. But what was the reason for it? Why did they not have characteristics that fitted together the same as the characteristics that we saw in, you know, flowering plants and in mammals and so on? Why? Nobody really knew. 1977. This is the front cover of the New York Times, and I think we can all agree we would all like to sort of be on the front cover of the New York Times with our feet up on the table and looking pretty cool. This is Carl Woese. And Woese's paper in 1977 was absolutely spectacular. He used ribosomal RNA sequences to say that in a single paper, he said a whole bunch of different things. So first of all, that we have a molecule that is present in E. coli and elephants. Okay, and this molecule it's homologous across all of life. Every cellular organism has it. It's a ribosomal RNA molecule. It's so important to the core functioning of a cell that all cellular life will have this molecule. And its sequence can help us to reconstruct the evolutionary history of the organisms in which it's found. He also said things about the progenote, what he came up with at the time. He thought there was a single origin. He said that there were three main primary domains of life, which were at the time were called archaebacteria, bacteria, and eukaryotes. He also said that the vast majority of the diversity of life on the planet was not eukaryote, it was prokaryote. The prokaryotes had the major amount of diversity. It's an extraordinary paper, front page of the New York Times. It changed so many people's lives. From there on, the ribosomal RNA sequence, the small subunit ribosomal RNA sequence, became the most sequenced molecule in all of science. From there on, the most sequenced molecule by directed sequences. Now we've got environmental sequencing of metagenomes and things, so it's a little bit different. But at the time, it's that. And I don't know how many sequences there are now, but there are probably millions. And it's all based, you can trace the origins of it to this particular paper. He constructed a phylogenetic tree of life. This particular tree is a little bit later. It's a few years later and has a little bit more details. But he said this is what life was like, and some rooting, put the root of the tree of life down, which split two of the prokaryote groups and put archaea closer to eukaryotes. I'll say a few things about this. If you feel there is a tree of life, then you build a tree of life. You use the data to build a tree. Okay? That's what the relationship between ontology and epistemology, right? If you don't think there's a tree of life, but you think there's a different model, that life has evolved in a different way, and I guess from the title of my lecture, I think that life has evolved both in this kind of way, but also very significantly by the merging of evolving objects with one another. And hopefully the data I present will persuade you of this. Richard Dawkins, who can give a talk without talking about Dawkins, he wrote in chapter 10 of The Blind Watchmaker, is an entire chapter devoted to what he calls the one true tree of life. And down here, if I can put a cursor over it here, he said the important thing about this system of rings. So he said that, you know, trees of life were like a system of rings, so groups within groups within groups, system of rings within rings. And it is that it is perfectly nested. And he italicized perfectly nested. He said, you know, with the implication that there was no exception to this. Never, not on a single solitary occasion will the rings that we draw intersect each other. Take any two overlapping rings, and it will always be true to say that one lies wholly inside the other, etc. So he said there is one pure, absolutely true tree of life. The thing I find about this is that this is several years after the Nobel Prize was won for transposons, which are jumping genes, which move genetic information from one species to another. It was known about when he wrote this. So I don't know why he wrote this way, but he did. But jumping genes will move genetic material from one organism to another in a very non-tree-like way. The issue is whether this is trivial in extent, whether it's limited to special categories of genes, whether it's limited to special kinds of organisms, or whether it's a universal truth. Okay? So what I would say at this point is I'm now going to go from, I started out with the Scala Naturae, moved to trees of life, and I'm going to move now on to networks. The hypothesis is that evolutionary history is tree-like. And I think for a period of about 150 years, this moved from being a hypothesis or a set of hypotheses of groups within groups within groups to where it became axiomatic, where it became an axiom, not a hypothesis that was actively being tested. It was an axiom, an agreed upon starting point. There is a tree of life. We will write software to understand it. We will put data into the software with the expectation that it will make a tree. And what I would say is that sometimes this is not a really great thing to do. I'd like to thank Charles Darwin for this quote here, because in fact, Darwin said so many things that you'll always find something that can help your cause. He said the importance for classification of trifling characters mainly depends on their being correlated with several other characters of more or less importance. So that is to say that if there is a great tree of life, then, and it really is the case, then all of the data should really start supporting that tree of life. We should be able to put the data in, and it will all come out and say, yeah, we agree, we agree, we agree. Congruence, consilience, all of these ideas that the data should all agree on this. He said the value indeed of an aggregate of characters is very evident. A classification founded on any single character, however important it may be, and for that I think ribosomal RNA phylogenies, which influenced a lot of the world, has always failed. We made a tree. This is in 2004. It's 20 years ago today. I had a very talented postdoc at the time, Chris Creevy. He did a lot of the work. Mark Wilkinson is down here in the audience as well. And Mark enlightened us on how to use super trees, phylogenetic super trees. These are meta-analytical trees. So we took 61 genomes, 1,117 gene families, and we said, now put them all together in a great tree of life. And this is what the tree looked like. It looks a little bit more like a comb than a tree. And the problem is that we couldn't get large-scale agreement across these trees. Any relationship you saw was almost always contradicted by some other relationship in some other gene. The genes would not fit onto a single unifying tree. These are all prokaryotes. I don't have the names on them here. Those numbers are just support values where some relationships sort of seen quite often, and some of them were, but we found no set of relationship that every single tree supported. None. This is 20 years ago. Okay, small data set by today's standards. Today we have 2 million prokaryotic genomes. At the time we had 61. We found no agreement, nothing really. So this sort of, to be honest, we set about doing this to make the tree of life using a super tree approach. And when we failed to make it, we published as it is in Proceedings of the Royal Society, because we couldn't find agreement between them. And so we started thinking, does this explain C.B. Van Niel's despair in 1945? Why are the different kinds of prokaryote and their characteristics not supporting a single classification scheme? Maybe it's because there isn't one. And maybe it's because horizontal gene transfer has muddied the waters so much. Since then, in the last 20 years, we have published a lot of papers on this. And I guess I'm standing here today saying that I'm pretty convinced that outside of some small groups here and there, pretty much there is no great tree of life. What there is a great network of life. But the mergers of evolving objects, by which I mean bits of genes, parts of genes, full genes, big sections of chromosomes, half of a genome, all the way up along across scales, these mergers have been super important in the evolutionary history of life. And furthermore, should we analyze this properly, it has one big advantage that the tree of life doesn't have, is that it can explain the data. Starting at scales, I'm just going to talk briefly about genes. When you actually look at genes, you figure out that they are actually a little bit like Matryoshka dolls. So we start taking a pair of genes, and if they're homologous across their full length, so unbroken, the two genes are maybe some mutations, but more or less they're the same across their full length. We'll draw a line between them, we'll start making a network. If they're not homologous across their full length and they've only got partial homology, then we'll color it a little bit more red, If they've got full homology, it's yellow. So it goes yellow, green, red. And this is what our networks start to look like. We start seeing groups like this. Every one of these nodes is a gene. Every edge is a statement of homology. In other words, a statement that they share a common ancestor of some kind at some point in their history. And the ones that are red over there are highly mosaic. The ones that are mostly green and yellow are a little less mosaic. But this is not a very tree-like document or a very tree-like image. But it is a view of the evolutionary history of life on the planet. There's 2,000 genomes in there. Okay. Each gene is compared to every other gene in the whole data set. And if they have homology across their full length, then we give them a yellow dot. The two are yellow. And otherwise, they become more progressively green and purple and all of those lovely colors until they get to be a much darker red. We then put them into communities and we sort of give them the average color of their community because otherwise we couldn't see it very well. But the point is that at the level of genes, we don't see tree likeness. Embedded within some of these communities, there will be some tree likeness, but overall, recombination, swapping of genes, merging of genes together, gene loss, partial gene loss has been a very, very important part of evolutionary history. Moving to organisms, and while I'm a microbiologist, we have done a little bit every now and again on what we call the outgroups, the animals and plants. Okay, that was a joke. I'm not very good at jokes. So this is a paper, I was only very slightly involved in it, but I put it up because I think it's interesting. Sequenced a bunch of polar bear genomes and a bunch of brown bear genomes. There's a timeline from top to bottom there of the evolutionary history of bears. They mostly separated from each other about 500,000, where am I with my arrow? Here we go. About 500,000 years ago, more or less, they separated, but you're finding several journeys of polar bear alleles, polar bear type genes into the brown bear lineage. And so what you're seeing here are hybrids. This is just one example, but hybrids and hybrid speciation, that's not new, that's very, very well known. But the point is that until we had genomes, a lot of this became hidden. It's not obvious. Brown bears look like a brown bear and a polar bear looks like a polar bear. It's not surprising maybe that Richard Dawkins said, you know, these are two separate things. But are they really if their genomes aren't? And the problem you have is that if every part of the brown bear genome doesn't trace back to the same origin, but some parts of it have to go through polar bear, then that's not a tree likeness. That's a non-tree-like event. And the tree of life doesn't predict that. It predicts that it should all trace to this ramification point here at 479,000 years ago, more or less. But that's just bears. This is work that comes out of Chris Jiggan's lab in Cambridge, and here they're looking specifically at genes that are responsive. Sorry, this is Heliconius, really good, nice group of butterflies, about 40 species, good species. When I say good species, I mean that 9,999 times out of every 10,000, when there's a mating, then the offspring, it's between two of the same kind and their offspring, and every now and again you will find hybrids form. But when we start tracing in the genomes where the genes are for, we can see the genes moving laterally over and back across what are otherwise good species, separate species. But these genomes move over and back across. This is a little bit old, 2021, but I think if you were to count the amount of DNA that's moving laterally across these 40, you might get very, very close to a whole genome's worth of movement. It's not the same for all genes. The genes that move the most are the ones for color patterning. They seem to be moving very, very freely. And what actually happens, just in case you're not, you've got two good species, they'll mate. The offspring probably quite often doesn't look great. It's not sort of terribly healthy, but it'll mate back into one or the other, bringing the genes with it, and then those genes will be in the other species. Ourselves, did Neanderthals ever really go extinct? If lots of Europeans are walking around with Neanderthal alleles, bits, chunks of genome that don't trace back to this common ancestor back here, the common ancestor between modern humans, Denisovans and Neanderthals, if it doesn't trace directly back here, but instead goes via some circuitous route, then we've still got a group of archaic humans, Neanderthals, that are not quite fully extinct. The most recent data on this, you can see the percentages of genomes that have moved. Those percentages keep going up. They don't go down, they keep going up. Okay. And there's some sort of great YouTube videos on that right now from the people that are doing this work. And of course, Svante Pääbo got the Nobel Prize a couple of years ago for this work. The origin of the eukaryotic cell, always a mystery, always a very unusual thing to be to try to explain. Ribosomal RNA phylogenies. If I go back all those slides, Carl Woese put it like this. This is what happened. Eukaryotes emerged from a group that predates the diversification of archaea. This is autogenous eukaryogenesis. In other words, the eukaryotes arose. Okay, they got this. The eukaryotes have got all of these inclusions, all of these organelles, and they just arose according to the tree. But when you start looking at the data, they didn't just arise. They formed by one of the most significant mergers in the history of life. And we're pretty clear, because we can see the evolutionary history of the two groups of genes, that the genes, quite a lot, come from archaea, and specifically these new archaea that we found over the last decade, called Asgard archaea and alpha proteobacteria. This was said by Lynn Margulis in 1967. She published a paper, and she's a very young scientist. She got absolutely crucified for this. For years and years. There's a paper in 1976 in Nature that was very, very stridently said that the mitochondrion, which Lynn Margulis had said was once a free-living bacteria, the mitochondrion was no more than an invagination of the membrane and a convenient place to put a bunch of enzymes to make energy for the eukaryotic cell. It wasn't until Carl Woese's group in 1987 or 88 published a ribosomal phylogeny where they could isolate the mitochondrial DNA and prove that it was from a free-living bacterium, or once free-living bacterium, that we now saw that eukaryotes are a true, I think, egalitarian merger of two prokaryotes, not the autogenous tree-like evolution, but instead the merger evolution. Lots and lots of papers on that right now. About 15 years, well, actually, sorry, what are we, 2024? 19 years ago, the word pangenome was coined for the first time. So, sorry, these are my colleagues. This is Mary O'Connell and this is Alan McNally, and we wrote this sort of slightly inflammatory article now about 7 years ago. I'll get to that in a second. First E. coli came out, right? So it wasn't the first bacterium that was sequenced, it was the sixth. First E. coli came out, and that's great. We all look at E. coli, we look at the genome, we go, these are the genes that it has. But you get to do something really great when the second genome is sequenced. You get to do comparative genomics. And that's really, really an interesting thing to do. What does one have that the other doesn't? You know, what mutations can you see? And then a third one came out. And when they looked at the first three, it's paper by Welch, they saw that in total, only 39.5% of the total number of genes that they found were common to all three. The rest were either found in two, or they were orphans and found in one. If you found three humans that only had 40% of their genome in common, you know, one of them would have to be an echinoderm human, and the other one would have to be, I don't know, whatever. You don't see that kind of variation. But yet at their core genome, these core genomes, you could line them up, and they were not quite identical, but they were very similar to each other. But the presence and absence of genes was rampant across these E. coli's. Okay? Tree-like evolution doesn't explain that very well. We move along, we sequence more and more and more, different kinds of species, different kinds of organisms. Some guys that were working on what's called the pneumococcus, they finally coined the term pangenome. And we sequence more and more bacterial genomes. And as we sequence now about 100,000 E. coli, was probably a lot more, we're finding that in what good microbiologists would call an E. coli, the total number of genes we find in there now is about 100,000. Though no E. coli has more than about 5,500. That's how much diversity there is in the E. coli genome. That's how much horizontal gene transfer and loss occurs in these genomes. I'll get onto it in a second. But the issue was then, why? Why do prokaryotes have pangenomes? I'm going to try and maybe explain this a little bit more. This is just a Venn diagram that's sort of explaining what's going on. You've got a core genome, you've got an accessory genome, and some species, this is nature microbiology, some species are closed. So Mycoplasma genitalium, for instance, is a bacterium that really has very little variation. There's about 3 or 4 genes that show presence, absence variation across the species. E. coli, 100,000. So it can be very, very different kinds of organisms. So not every organism is the same. E. coli has an enormous, what we call long-term effective population size. I don't want to get into the technical details, but if you've got a big effective population size, then natural selection is very efficient in that species. Species where natural selection is inefficient are small, have a small effective population size, and random genetic drift dominates from one generation to the next, okay? So it's just a point about population genetics. So E. coli, with its massive long-term effective population size, should be very, very efficient when it comes to natural selection. Now, I'm going to just talk a little bit, first of all, about sort of pangenomes, and I'm then going to try and persuade you why they exist. So this is kind of one of these figures during World War II in what's now Columbia University in New York. There was a group set up by the Americans called the SRS, which is the Statistical Research something Society, maybe. And the head of that was a guy called Abraham Wald. Now, Wald was a Polish Jew who, you know, when Nazi Germany was sort of, you know, turning Europe into a really terrible place, fled to America, and Wald was quite a genius, really. He was a remarkable person, but he was set up as the head of this place. Now the statisticians and mathematicians in the SRS were phenomenal. Several of them won Nobel Prizes for various reasons. But the army came to him and they said, what we want you to do is to do a statistical analysis on the bullet holes that we find in airplanes that have come back from the European theater of war. And what you need to do is, I don't know much about war, but, you know, is you need to put armor in those places that are more likely to be hit. But you don't want to put too much armor because the plane is too heavy and it won't take. So they said to Wald, find the places that are most often hit and we'll armor them up a bit more. But Wald sort of flipped the whole logic on its head and said, actually, no. The planes that come back, we need to reinforce those places where there are no bullet holes. Because the planes that got hit in those places, they didn't come back. Right? And so if you swap bullet holes for presence, absence, variation, and so on in prokaryote genomes, or in any kind of genome, mutations, where you see no mutations, there's no mutations there because there can't be. Because the organisms that had a mutation there, they didn't come back. Okay? So when we started sequencing bacterial genomes, we expected to see variation. You know, every zoologist, you know, sort of works on variation. Look at, you know, leg length in giraffes and you know, what have you. Right? Variation is what we expect. When we started sequencing bacterial genomes, we didn't expect the kind of variation we saw. It's sort of like as if the bacterial genomes that we saw were rifled with things that we didn't expect them to see. They were barely hanging together, and yet they worked very, very, very well. So how do you explain this enormous amount of variation that you don't see in animals, that you don't see in plants, in terms of genomes, in terms of gene presence and absence? There's a lot of genomic variation among the junk DNA in animals and plants, I'll just say that, but among the protein coding genes, you don't tend to see it. So why do we see this? Could it be that purifying selection is the answer? Well, purifying selection would eliminate new gene combinations. Whenever a new gene hopped into a genome, purifying selection would say, no, thank you, I don't want that. So purifying selection can't explain it. What about random genetic drift? Are these genes hopping into an E. coli, but if we sequence the descendant of that E. coli next Tuesday, those genes would hop back out again? They're having no effect. Well, that seems very, very unlikely given the large effective population size of organisms like E. coli. They're very, very efficient. They don't have any junk DNA. Very, very little. They might have small amounts, but they certainly don't have the kind of amount that we see there. And it can't possibly explain hundreds of thousands of genes. And so the answer we've proposed is that there's positive selection on new genotypes. That in fact, all of this variation, the dominant influence on it is positive selection for new kinds of organisms. And that would suggest that on this planet, the size of niche space is enormous, particularly if you're a prokaryote. Okay, and this is just a technical detail, sorry. So where does that leave us? And what do we think? And why is that important? Well, there's a few reasons I'm going to suggest that it's important. First of all, if random genetic drift dominated bacterial genomes, then the bacterial genome that we sequence, lots of those genes wouldn't be important. They wouldn't contribute much to the fitness of the organism, but yet we know that they do. So what's emerging now is that pangenomes are in fact a kind of ecosystem on their own. And the genes like hanging out with other genes and they hate hanging out with some genes. And not just that, but in the same way that ecosystems, if you go to the savannah, if you go to the deep sea or wherever, ecosystems have a kind of a hierarchy. They've got interactions in various ways. I think pangenomes have something very, very similar. So we set about asking this question. The so whatness is that drift-dominated pangenomes could mean that evolution is not predictable. And that's an interesting thing for us. How predictable might evolution be? So if I'm getting rid of the tree of life and replacing it with a network of life, it's got to have some usefulness as well. Okay? So if drift dominates prokaryote evolution, then the genomes that we see don't really matter much because these genes are just hopping around the place and they're not contributing much to fitness, and pangenomes don't have rules. Otherwise, if it is selection dominated, it could mean that evolution is somewhat predictable and pangenomes might have rules. And so the interesting thing is that selection dominated evolution should leave traces. We should see sets of genes having preferences for appearing together and sets of genes having preferences for not appearing together. In other words, that the genome itself would have some kind of rules. So setting about this, we started looking at the co-occurrence and avoidance of genes. And Fiona Whelan, who's now in Manchester University, but she worked with me for a few years on this, trying to understand pairwise presence-absence covariation. We can build networks of genes, then different kinds of networks to the ones that I was showing you earlier. And here we're going to link two genes together if they're friends. It's kind of like the Twitter or the Facebook or the Instagram network. So two genes are hooked up to each other if they like to hang out together. We got some statistics for constructing these. And so we can construct networks of association where genes show a surprising amount of association with each other, and networks of dissociation where genes really, really don't like to be in the same genome as well. And this, I think, has evolved over the last 4.5 billion years to be a situation where particular genes like to be together and particular genes like not to be. Here's an example from Pseudomonas aeruginosa, which is a bacterium that you find quite often in the lungs of cystic fibrosis patients. And these are about 200 genomes that all come from cystic fibrosis patients in Canada. And we're just zooming in on what this looks like. And the colors aren't fantastic on this here, but you can see two different groups of genes, the light pink and the dark pink. So I must change the color on that. But where you see, say, for any of these two groups, if you see one, then you're highly likely to see the others in that group. Okay, but also if you see one group of these, then you might be less likely to see the other group. And sometimes we've seen where they really, really don't like to be together at all. This is a paper from January this year, so I'm really getting closer to the end. So Alan Bevan and Maria Rosa Domingo Sananes used to work with me, and we published a paper called Contingency, Repeatability, and Predictability in the Evolution of a Prokaryote Pangenome. So contingency, historical contingency, is an idea that Stephen Jay Gould came up with. And he wrote about it in his book, The Wonderful Life, The Burgess Shale and the Nature of History. And what he said, that any replay of the tape of life would lead evolution down a pathway radically different from the road actually taken. That's a very strong statement, really. I think that he's saying that if we reran the experiment of evolution on this planet, you wouldn't see the same thing. I think he's right in many ways. Of course, you wouldn't see the exact same thing. The people in this room would not look exactly the same, but lots of things I think can arise. And Stephen Jay Gould was another person who wrote a lot of things, and it's not entirely clear what he meant by this statement, but we're still having a look at it. The question is, if you take a big diversity of E. coli, where, as I've said to you, all their genomes can be really, really different for each other, and we start looking for genes that hopped into different groups of E. coli, and we ask that we only look at the genes where that happened at least 8 times. So we're putting a gene, not us, but nature put the same gene effectively into a bunch of different genetic backgrounds. What happened next? And what we could do, using a method called a random forest, I'll get to it in a second, was we could ask what happened next and whether similar things happened after these genes came in, whether they attracted the same sets of genes into that genome, whether they repulsed the same sets. In other words, was it predictable what would happen next? We started seeing things that we called mutualisms, commensalisms. We saw competition, genes that refused to be in the same genome with each other. And we think that we've seen predation, parasitism, etc. These are all words from macroecology that now I think can be applied to genomes because there isn't a tree of life and because these genes continuously meet each other in a way that's analogous to animals and plants meeting each other in a macroecological setting. This is just a diagram talking about a decision tree. It's where this is how it worked. I'll very briefly say this. We pick a gene, gene A. We split our genomes. We've got 2,500 E. coli genomes when we do this. Those that have gene A, those that have not. Now we pick a gene, gene B, and we split the data set again, and gene C and gene D and so on. And we ask, is there any path down along this decision tree that can strongly predict for us the presence of gene X or the absence of gene X? Okay. And there's an algorithm to do it. It's been well worked out for a long, long time. And it turns out most of the time, no, but a lot of the time, yes. We can predict whether gene X is going to be present or absent quite a lot of the time. This goes into what's called a random forest. But I'll put it like this to you. If I gave you an E. coli genome, stack of E. coli genes, and I said to you, hide one of those genes behind your back, and then give me back the rest of the genome, I'd have a very good chance of telling you what that gene was. In other words, I can tell from the rest of the genome what that gene is. Okay? Not all the time, but quite a lot of the time. 30–40% of the time. I expect to be able to do that, if I was to guess randomly, 0.001% of the time. In reality, the algorithm will do it about 30–35% of the time. 2,500 E. coli genomes is a very small number of the total number of genomes we could have. We build up a network that looks like this, and these are all genes that predict other genes. This is what this particular network looks like. And this is the network of significantly predicted genes and their predictors. I'll just go through what some of them are like. We're given the genes, the names K, J, K, L, and M, and the red T's means they significantly avoid. And the green arrows mean they love to be together. In fact, actually, L and M are always together. And K and J, they like to be together, but J likes to be with K more than K likes to be with J. It happens. But it turns out that J and K and L and M make these antiporters. They're pumping things in and out of a membrane. Okay? And if you put the two of them in at the same time, while one group is pumping in, the other group is straight away pumping out. It's useless. So you can't have the two together. So the pangenome will evolve these kinds of things where these two genes, these two sets of genes will not appear together because there's no point. Because I put it another way, the fitness effect of having the two together is really, really negative. And so any genome that has these together, it's not going to last very, very long. Here's another set of genes that like to appear together. A, B, and C are always together. These are the red ones. This is across the 2,500 E. coli here. The red ones, always together. D, the sort of purpley one, when it is present, it predicts the presence of the red ones, but not vice versa. Is that okay? Does that make sense? Does that pattern look like it's working? Okay. E will predict the presence of A and B, but not vice versa. But F and E really avoid each other. The presence of one is a strong predictor of the absence of the other. Okay. G strongly avoids E as well. And G, H, and I quite like to be together in these genomes. That's kind of what the pattern looks like. So just to pause there, because I'm actually doing well on time. What this means is that pangenomes are selected. What does that really mean? It means that selection dominates. There's room for drift, there's room for population bottlenecks, all of these other evolutionary things. But it might mean that genomes are predictable. And the next thing it might mean is that pangenomes might be a language. So this is a bit of a handbrake turn into artificial intelligence for the last 3 or 4 minutes. So for the last few years, I've turned my lab entirely into an AI lab. So have any of you used ChatGPT, or you've at least heard of its existence and so on, and Claude.ai and so on, and the world has gone absolutely mad. We've been asking the question whether a pangenome therefore is kind of like a language. So in language, you won't find the words xylophone and zebra in the same sentence. Well, outside of sort of creative writing, maybe you might find it, or you know, that sort of thing. But generally not. Some words belong well together. Are you okay to do a little test? Okay, what's the next word in the sentence? I'll give you 4 choices and you tell me your favorite. There's 30,000 words in the English language. I'll narrow it down to 4, but I want you to show of hands to see which one you want. The word is finish, and I want you to tell me what the next word is. All right? Finish first, finish last, finish up, finish down. Okay, finish first. Show of hands. Okay. Finish last. Finish up. Finish down. Okay, I'll give you a little bit more information. Nice guys finish. Last. Everybody, right? Context. And so this is very, very similar to a genome, I think. The context is now emerging that evolution has created this kind of context in genome. That genes prefer each other in the same way that words like to be together in languages. So we've developed, I wrote this myself actually, a program called PanGPT, which is a generative pre-trained transformer software for building GPTs from pangenomes. It will only work if genomes are selected, because natural language is actually selected, because it's selected for meaning. It will not work if drift dominates. We've been working on this for a while, and if any of you know anybody in the BBSRC, tell them to fund our stuff, please. And this is just some of the code, and so we're working on this. But a gene has a remarkable amount of similarity, or a genome has a remarkable amount of similarity. In language, you have something very similar to genes. So if you say unexplainableness, it's not really a common word, but un is a common prefix. Explain, or X might be the next part of a word. Plainableness, okay? Genes can be broken up the same way into promoters, operators, protein domains, terminators, and so on, and regulations. And the relationships between them can emerge as well. And I think this is the correct way to start thinking about and looking at these kinds of organisms that are engaged with a lot with horizontal transfer, because I think that the similarities between the two are very, very interesting. So I've trained what's called an LPM, which is a large pangenome model, starting off with our 2,500 E. coli's, with a vocabulary size of 57,161 different kinds of gene family. I've had to exclude some things in there. So if this is English language, we don't break the English language when we're doing LLMs, large language models. We don't break it up into the words, the 30,000 words. We break them up into what are called tokens. Sometimes a word is a token, but something, like I said, unexplainable. You break up that word into several tokens. We haven't done this here because I've got funding pending to do this. We have a vocabulary size of this. We trained it for what are called 22 epochs, and after about 40 hours, this happened. It turns out, so all this graphs mean is that the software learned the pangenome language. It got better when I did that test that I've just done with you guys. You know, nice guys, finish. You do this test continuously with your language. At the beginning, the pangenomes, it didn't know what should come next. At the end of this, it had a very good idea. It was getting about 75% of it correct, what should come next. We can learn the language of pangenomes, and it's entirely because they've evolved via a network. This is just some more information about it. It doesn't matter. Take home message, last slide, you'll be pleased to know, and this is the only thing that's between us and a glass of wine. Selection versus neutrality debate matters. The tree likeness versus the network likeness debate matters. The explainability of evolutionary history matters. Into the future, if I was to talk about the future of evolution, it's unquestioned that we're going to use AI, we're going to use synthetic biology, we're going to use genome editing. If that doesn't have rails, if that doesn't have protections, I'm not really sure where it's going to go. It has some rails right now, but I think it's very, very important to be able to say that the merger, the last merger I'll talk about, is the merger of AI, genome editing, and genomics. And the future, the next 100 years, will involve a lot of that. And it's important for us to realize that because we can put genes from one organism into another and predict what they're going to do, that very, very quickly, a large pangenome model will make 1 trillion genomes in a morning, and you'll be able to select which ones you like and go into the lab and make them. It's a new world, and it's one that we need to be very careful of. The evolution of bacterial genomes are predictable in some ways, and this can open a new door to evolution-inspired AI genome analysis and design. Was the last. It can have tremendous benefits for vaccinology, for dealing with antibiotic resistance, for generating new kinds of energy. Okay, we can predict how to make a genome that would produce new kinds of aviation fuel if that's what we wanted, to decarbonize the planet. We could think about how to make genetic constructs to do that, because with these kinds of models. But as we all know as well, they need to have rails and we need to have safety. Come to the end. I feel I've ended on a downer there. Thank you so much for listening to me. I hope I didn't go on too much about things, but I think I'm just about in time. Thank you so much.